gentlemen, welcome on stage. You have your microphones right there, and feel free to start.
To be at this session with a fantastic panel. We have myself in first to speak about the view of a medical oncologist of the progress and the way to move. Marc is a statistician, well known, and he speaks about new methodology of statistic. We have Samer El-Bawa of the Servier company to speak about, you will see the view of the pharmaceutical company. We have also Cyril SARRAUSTE De Menthiere. It is a patient, it is a view of the patient. And we have also Federico GOODSAID about the Ariana Pharma for artificial intelligence. I will begin by, I think my slides are there, if it's possible. I don't know if I have to, it's work? Yeah. Donc, I have a double institution there. I work in Saint Antoine Hospital in Paris, Department of Medical Oncology, and I also president of a foundation, the ARCAD Foundation. It's a foundation for GI cancer. We'll take an example about the colorectal cancer. The colorectal cancer, the progress was huge for metastatic disease since 25 years. When I began medical oncology, it was with Marc, with folinic acid at 5-FU, and the median survival was 12 months at this time. And now we are, like you will see, it's really depending of the genes of the tumor. And we are now around 20 and 40 months. It's a huge progress. And really, it's like all tumor. The first one was the lung cancer, with a lot of niche of therapy, with really some with molecular signature, with immunochemistry for some other marker. Huge progress, and you have the chance to coordinate the study of the KNOT 177 in MSI colorectal cancer, with a huge difference in PFS and improvement in overall survival with time. And it's really the first time it will be possible to have a long control of the disease. It is for MSI 34 patients without progression at 5 years. In the first time, we have the hope to cure a metastatic disease in colorectal cancer with a medical therapy. There it's anti-PD-1, and it's now a lot of other group of patients with less benefit, but with important benefit. HER2 and the mutation of RAS, the mutation of now the GOODSAID with a New England paper, Lancet paper, every not months, but every it's 2 or 3 by year, and like lung, we move on. In adjuvant setting, it's really more complex, because in adjuvant setting, we try to decrease the risk of relapse with adjuvant therapy, and it's not a lot of progress since a very long time, because Mosaic, the study with oxaliplatin, the only drug after 5-FU to improve overall survival and DFS, was 20 years ago with a little benefit. And the problem is we treat a lot of patients for a very little benefit, with a lot of toxicity for the totality of the population. And it was also for 5-FU, for only stage 2, the risk of relapse is very little, and we have to have chemo for a lot of patients for a very little benefit. And a lot of drug was tested without any benefit because it's very difficult in adjuvant to improve the DFS, and the only little progress was to have the opportunity to decrease the duration of chemo on some patients. And the hope is really the ctDNA, and it's now a new area. We have a New England paper by the Australian team of Thai for the proof of concept. We have the Japanese paper in Nature Medicine with also a proof of concept, and all the future will be with ctDNA to select a population with risk. And you have on the curve the fact that at the left you have sorry, I will go. I don't have a pointer, but you see that if the patient didn't have ctDNA, the risk of relapse is very low. If patient had ctDNA, the chemo works and decrease the risk of relapse, but it's not randomized trial and it's not in clinical practice at this time, but all the development now of adjuvant therapy in colorectal cancer will be with ctDNA. To close my presentation, just to focus on the ARCAD Foundation. The ARCAD Foundation is a foundation now 15 years old, and we have collected all the, not all, but 60 phase 3 around the world concerning therapy of metastatic colorectal cancer, academic or industrial. And the goal is to improve the approval process. It's a very difficult task, but we published 25 papers with this database, and the database in Mayo Clinic and increased with the time. And really the goal, the dream is to avoid randomization against a placebo, and it is to have a synthetic control arm for refractory metastatic colorectal cancer. And we have published a letter in Nature Medicine to prove it's probably possible. And really, it's a pairing with a patient of the database and not randomization, because randomization for a placebo is very complex in medicine and in oncology. And this work is ongoing, a lot of work to move on, to work with company, to work with agency, to have this opportunity. Thank you very much for your attention. And now we are speaking about statistics. Hop.
Thank you, Thierry. Thank you, Thierry. As Thierry reminded me, we've been doing clinical trials for a long time. And one of the frustrations when we design or analyze clinical trials is that, as you know, we have a primary endpoint which is pre-specified in the trial. And if this primary endpoint doesn't reach statistical significance, we are not able to say anything about the other endpoints in the trial. And that is just wrong. It's wrong. It's not good science. It's not good for the patients. It's not good for statistics. So what I'm going to show you today is a new statistical method that combines several endpoints in a clinical trial and allows us to move beyond a single primary endpoint, which I think is well overdue. So this is my affiliation, and here is the background of this trial. I'm going to look at a specific example of this in the context of patients with head and neck cancer who are treated with radiation therapy and cisplatin. And these patients, about 70% of them, develop a very bad toxicity called severe oral mucositis, SOM. Now, WHO grade 3 SOM is patients who have severe pain, and they can often not eat solid food, so they have to be drinking food instead of eating it. And then grade 4 is even worse. Patients oftentimes who have grade 4 cannot eat or drink, and they have to be intubated. So it's a really bothersome toxicity for patients. The traditional endpoint for clinical trials that look at this specific toxicity, according to the Food and Drug Administration, the FDA in the US, is the incidence of WHO grade 3 or 4. That means the proportion of patients who have this severe oral mucositis. But when you think of it, is it sufficient to look at incidence? Well, it's not. And we can use a new method of analysis called GPC, generalized pairwise comparisons. Then we look at patients who have grade 4. And we look at 2 patients, one from the control group, one from the treated group. And we look at which of these 2 patients had a grade 4 sum. If it's the treated patients, that's bad. That means the control was doing better. If it's the control patients, it's good for the treatment because the treatment did better than the control. And if both patients had grade 4 sum, that means it's a tie. And then if the patients did not have grade 4, we look at whether they had grade 3. So again, we look at the same pair of patients and we look at grade 3. Suppose they both had grade 3. Now we look at duration of sum, because it's not the same to have this sum for 1 day or 2 days as opposed to having it for 1 month. It's completely different from the patient perspective. And finally, if we have 2 patients who have the same grade, the same duration, then we look at the time to onset of the sum, because the later they get it, the better, because that means they will have received much of that radiotherapy before they develop this toxicity. So it's a completely new way of analyzing the data, where we don't have a single endpoint, we have 4 prioritized outcomes, right? And we look at them in succession. So here is the analysis we did for an American drug company. They developed a drug called avazopresine that in fact prevents the occurrence of SUM, or at least reduces the likelihood of a SUM. And this is the result of a trial they performed. You see here the results of the trial. 166 patients were randomized to placebo, and 241 were randomized to receive avazopresine. And as you see, 64% had grade 3 or 4 SUM in the placebo group versus 54. So that's a 10% benefit. But that's a 10% benefit in terms of incidence alone, regardless of the duration, regardless of the time to onset. We reanalyze these data using generalized pairwise comparisons. So what you see there is these little people represent the proportion of pairs that are better for avazopresine, better for control, or neutral ties. And as you see, we look at incidence of grade 4. 26% had grade 4 in AVA, 16% were better in placebo, and so that means a 10% difference, 26% minus 16%, it's a 10% net benefit in favor of the drug, of avazopresine, right? So that's 10%, and the p-value for that is 0.04. Now that would be the sort of traditional analysis looking at just grade 4 sums. And what we can do then is for patients who did not have grade 4, did they have grade 3? And then we have an additional 3% benefit. For 2 people who had grade 4 or grade 3, how much did it last? What was the duration? 10% versus 6%, that's another 3% benefit. And so we go down the list of prioritized outcomes, and for each outcome, we add the benefit from treatment that is due to that specific outcome. So we end up, at the bottom of the slide, with a total net treatment benefit of 19%, as opposed to 10% upstairs. And instead of a p-value of 0.04, we now have a p-value of 0.001. So we've increased the statistical significance of the test. The p-value is much lower with a new method. And in my view, it's also much more clinically relevant for the patients. So it's a method that increases both statistical significance and clinical relevance. So I think it's a method that should be increasingly used. Coming back to the examples that Thierry ANDRE showed, when we design a trial in colorectal cancer, we've got to make a choice. Are we looking at survival as the primary endpoint, or progression-free survival, or maybe response rate? And then once we have made that choice, if that endpoint fails, typically we can't look at the other endpoints. And again, I can only repeat what I said, that is not an optimal approach. I think we can do way better by using this technique. And that is the end of my talk. Thank you very much.
My presentation is about the tools that we have at Ariana, how those tools are being used today, and how we anticipate they will be used in the very near future. When we have a population of patients, a starting population of patients in a clinical trial, we like to look for those patients that are likely to benefit the most from a therapy, and that's the foundation of precision medicine. And to do that, we use standard statistics to identify the population that responds to the therapy and the population that does not respond to the therapy. And the way we do that is by identifying the biomarkers that are capable of doing this. This looks a lot like a Maxwell demon, and if you're familiar with what a Maxwell demon is, it's capable of really identifying selectively those patients that will benefit from a therapy. So that's what you have when the patient population is large. When the patient population is small, if you use standard statistics, there are several reasons why you run into problems. The first one is because you start getting bias in the selection of the population that you're using to determine the biomarkers. The second one is because you start getting heterogeneity in that small population. And the third one is that, and this is probably the most important one, even if the first two did not happen, you have several statistically equivalent answers to what is the best biomarker to use for separating the two populations. And you end up with the wrong answer as to what that biomarker should be. So it doesn't work very well. At Ariana, yes, we can separate the two populations by identifying the best biomarkers that will be capable of doing that separation when you have a large population to start with. But it also works just as well when you have something like 20 patients in a clinical trial. And we've shown that for Alzheimer's at Anavex and for type 1 diabetes at IMSICE. It really works. It is a big difference. I'm not going to go today into how it works, but let's just say that the background to the success of this technology is in and of itself quite interesting. So we are capable of identifying those patients that are likely to benefit the most from a therapy. 20 years ago, when I was at the FDA and we were starting to talk about personalized medicine, the question came up, why is it that we're so interested in just efficacy and no one seems to be talking about similar patient selection biomarkers of safety? And it hasn't really changed too much in the past 20 years, but that might be about to change. And the reason why this is the case is because while most companion diagnostics, as of today, are all to identify patients that are likely to benefit from the therapeutic response, there are no safety companion diagnostics. The PIK3 drug class had a patient selection efficacy biomarker with it, and that's how patients were selected to receive the therapy, because you could select patients that would likely benefit in terms of the efficacy of the therapy. And then something happened after the approvals by the accelerated path at the FDA. There were multiple drugs that were approved in the class over the past 10 years or so. And those drugs, after accelerated approval, went into their confirmatory trials. And that's when something happened that might make us change a little bit how we look at precision medicine. There was an ODAC meeting set up by the FDA last year to examine the confirmatory trials for all of these drugs. And the results were something that really brought pause into the medical reviewers at the FDA. because there were decrements in overall survival in 6 randomized confirmatory trials. There was the same pattern observed across multiple trials. There was a higher rate of death due to adverse events in this PIK3 inhibitor arm. The decrease in overall survival was due to toxicity, and basically the whole PIK3 class has been affected, effectively killing the class. The response by the FDA has been Project Optimus, which is designed to optimize the dose to try to mitigate the effect that was observed for PIK3. And of course, in doing so, you're going from maximum tolerated dose to minimum efficacious dose, and there's a price to pay for that. What if we started thinking about patient selection biomarkers of safety? What if we could identify the biomarkers, the variants, that might be associated with an increased likelihood of death in these clinical trials? That idea was shared with the FDA, and Ariana and the FDA have set up, in fact, beginning this week, a challenge to identify patient selection biomarkers of liver safety. We're focusing on the liver because it was one of the most important PIK3 toxicities. And it's a very simple idea. And as you can see where Ariana's technology will be absolutely critical for this to work. There's a certain number of patients who died. There's a certain number, a much larger number, much larger number of patients who survived. And now we need to identify the biomarkers that will be able to separate those two populations. It's a fantasy at this point. We'll start working on this over the next few months, but it would be fantastic if we were able to identify any variants that might have preferentially been observed in the patients who were not able to survive this trial. And that's it. Thank you.
So thank you for the invitation, and I will be happy to discuss with you today about the integration of those new solutions, approaches, AI, machine learning in pharma research and development. So before starting, I would like to basically start with 2 points. The first one is to remind ourselves about what is the objective of all these new approaches, which is, let's not forget, is to bring safe and effective drugs to patients faster. This is really the key point, and we need to keep that in mind. The second one is, I just pulled out 2 papers, one from cancer associations, if you want, and the other one that came from FDA. It's a discussion paper about the use of those new solutions, if you want, in drug development and drug discovery. So it gives you a sense that this is really an important topic, and associations, regulators are also looking into that very seriously. We at Servier, we agree with that, and we believe actually that data and AI approaches are transformative science because they are going to impact the whole R&D value chain. On this slide, you can see different areas going from research up to clinical development. And I just highlighted examples of where we see already AI, machine learnings are already having an impact, or where some applications are already ongoing. If you look at research, I mean, obvious fields are, target identifications, and I will highlight one example of what we are doing at Servier in a second. Drug design, of course, those are very prominent examples, in addition to many others. As we move up to translational medicine space, we can think about the first human predictions in terms of dose or pharmacokinetics, for example. but also applications such as disease modeling that we believe is going to be really important. And again, I will highlight this in a second. As we move up to clinical development, of course, those approaches are already used to help in clinical trials design. So simulations are used not to predict the outcome of the study, sometimes it's tried, but more in order to help refine and define the way we are doing the phase trials. Another example is to find the patients with the molecular characteristic at the different centers. So those approaches are already used in R&D. So, you know, I want just to highlight a couple of examples in the research space and translational space. This is from Servier, but there are, I'm sure, in many other pharma companies, they have similar approaches. So since years, actually, at Servier, we have been generating a lot of knowledge in the research space, and we have built an in-house engine that we call Patrimony. And I mean, you can look at the paper where we describe more details about what is there. It's kind of knowledge-based tool, and we are using really this tool, you know, in our standard day-to-day work. So we are using it to do target prioritization, of course, but also drug repurposing, especially for pediatric cancer, where we have a special focus there. And in oncology, you know, every drug will go in combination at certain time. So we are using those in silico approaches to predict potential combination partners. Of course, we are using machine learnings to do, as I mentioned before, target validation in different diseases, or clustering of some diseases, so that we can better understand, you know, the molecular characteristics of the specific disease. Another area where we believe it's really in the translational space. Oh, it's not moving. Oh, one back. Okay, I'm missing one slide, but it's fine. Another area that we believe it's going really to be important is what I call disease modeling. Disease modeling is going to help us, you know, by leveraging all the data, biological data, patient data that are generated today, genomics data, proteomics data, clinical data, in addition to those approaches that we talked about, machine learning, etc. So the merger of those two things is used to establish disease models, and those disease models are going to help us really to have a good and deep understanding of the biology. We believe this is going to help us implement the precision medicine approaches that we would like to implement in our trials. With that, I'm going to stop actually here. The key message is those new approaches are already, but likely going to transform the way we discover and develop our drugs in the future. So, thank you.
I think it's important to have the point of the patient. Cyril is there for speaking about the fact that in the decision making now, it's a lot of progress with the medical oncology, the statistics, the artificial intelligence, the pharma, but the patient play an important role in the process, because the patient is there not only for cure, but also to participate to the decision of to help the agency, to help the company, to help at each part of the development. Cyril, maybe you can have a word.
Need another mic? No, okay, it's okay. Thank you so much. Yeah, I'm here as a patient. I'm co-responsible of one of the fourth section called Mon Réseau Cancer Colorectal from the association Patients en Réseau, who works on cancer of lungs, breast, and gyneco, and the last one, colorectal. So it's not so easy to give a point of view of patient after this 4 talk, but it's a very famous subject, and better than to have words from anonymous patient, me. I found on Twitter and LinkedIn this morning 2 beautiful posts that really express what we need as a patient. The first one I will try to translate is coming from the Hôpital Universitaire de Genève by the professor Pataki. Medicine is there to cure people with disease, and this medicine has to be efficient, and this medicine has to take in account the expertise of the patient and integrate this expertise in the healthcare process. So that's what you say. All the system, innovation, healthcare journey teams have to listen patient and what patient have done and understand, and so now it's an expertise during their healthcare journey. Because the other post was from, it was a slide from the ESMO 2 days ago, and it says that patients are never ideals. We are not in good shape, with a good mind, with a good health, of course. And so their context is messy and unpredictable. Treatment itself has many unknown factors. The only solution is more organized collaborations between the medical community and the patient community. And that thing, these few words summarized with these 2 posts, is really the real life, the reality, the real world evidence that all system, innovation, from innovation or for clinical or from fundamental research, have to take into account. And for that, we have to, the patient was a subject of study, the patient was centric, and now the patient is a partner, a collaborator of your job.
Thank you, Cyril. I think you tell that collaboration is important, but the panel there, it's a panel of all people at to work together to improve the therapy of cancer, and the medical oncologist, the statistician, but artificial intelligence is now a new step to understand. The biomarker is very complex as methodology, statistics, or by intelligence artificial. Maybe can really find a group of patients with better efficacy. And Pharma is really important because Pharma develops the drug, and it's really the collaboration between all together that it probably improves the things. And yeah, the things move very quickly and need new technology for evaluation, and really it's a crazy time for cancer. The improvement is in part huge, but a lot of patients died of cancer, and each year we find a new population of patients with pretty good results, and it is in relationship with the collaboration between all of the partners. Maybe some question in the room for the panel. Alain, maybe you have a question.
No, I think it's very nice to see on the same podium a patient, a treating physician, a statistician, a representative of the pharma industry, and a representative of let's say, new tools like artificial intelligence. And I'm sure that altogether you can push the agencies, the FDA, EMA, to change the way they are viewing drug development and registration. But I was quite disappointed. I was not in Madrid at ESMO, but I know that there was a symposium led by one famous Canadian oncologist who still conclude that the only reliable endpoint is survival. How do you react to this? And before having agencies changing their mind, one of the key stakeholders, if one of the key stakeholders is still thinking that the only way to assess drug is survival, it's quite disappointing.
Yeah.
It's a little bit, it's not true that it's not, it depends on the situation. You know that in colon cancer, DFS is the endpoint for the agency for adjuvant setting. In some, it's quality of life. It's sure that OS is the gold standard. But now it's really changed, and we have to prove that in a lot of situations it can be changed, but it needs a lot of work and need the statistician to prove that, to have a surrogate endpoint, and it's a long way. But the first we did together with Dan Sargent in Mayo Clinic was the correlation between DFS and OS. It is the goal of the ARCAD Foundation to have the database to try to change the endpoint and to have a surrogate endpoint. And it's not true that OS is only now the endpoint for a lot of trial. And Marc, maybe you can comment.
Yeah, absolutely. If you look at the number of approved drugs, at least in the US, many have not been approved based on OS, but PFS or sometimes even complete response or even response. So I don't think it's true at all that survival still dominates. Of course, it's the most important thing that everybody would want to improve. But a drug could be approved even if it doesn't prolong survival for good reasons, because you cannot deprive patients from that drug. So it's very difficult to show a benefit on survival if patients are allowed to cross over from the control treatment that they received to that new drug. So I think it's a bit naive to say, well, the only thing we care about is survival. It just doesn't work like that. And I think it would be counterproductive to do that. From a patient perspective, it may be different because survival is what matters, for sure. But I think for drug development, we have to look at many endpoints, including quality of life, by the way.
Survival is very important if you have the quality of life.
Yeah.
Survival with a bad, a worse quality of life doesn't matter, really. Most of people has to change their treatment, has to change the world they are living because they don't like it. For example, Professor Andre called about colorectal cancer. So sometimes we speak about ostomy. Some people ask to, and some, not definitely ostomy, some people ask to have a definitive ostomy because the quality of life is better with an ostomy, the artificial anus, is better with an artificial one than to try to be reconstruct for the digestive way. So the most important is really the quality of life. And it's incredible to have this question, do you prefer to live shorter but better or longer but worse?
Yes, the reason the patient is more and need more to be in the decision making. It's two different. We have FDA and we have Europe. We have also Asia, but the situation for approval is really different between US and Europe. And after it's the problem of reimbursement of the medication very different. And it is more open in US with the system, probably every know, that in Europe, where it's the first step is EMEA, and after it is a reimbursement by country, and it makes the things totally crazy sometimes.
I think everyone would agree with that, but on the other hand, you would not propose that a drug class be approved even though it actually causes more deaths than the standard of care? No, right? Okay, because that's where overall survival comes up.
Yeah, it's the topic to also try to understand better a trial like you demonstrate by the fact a drug can be efficacious but add some toxicity, and to try to find the good drug for the good patient, it's also very important. I think we have in time. It's time to close the session. It was a pleasure to have this session with you, and hope it's a view of the oncology right now with the panel, and thank you for your attention.